हाय एवरीवन फ्रेंड्स डू यू नो एक छोटी सी प्रोटीन फोल्डिंग बना देती है प्रोटीन को हीरो या फिर विलन एग्जैक्टली अगर प्रोटीन सही से फोल्ड हो जाए तो उसका फंक्शन परफेक्टली एक्टिवेट होता है जैसे एंजाइम्स सही तरह से काम करते हैं हॉर्मोन्स हैं एक एग्जाम्पल इसका हीमोग्लोबिन भी है लेकिन अगर प्रोटीन मिसफोल्ड हो जाए तो प्रोटीन अपने फंक्शन सही से नहीं कर पाता प्रोटीन बन जाता है एक विलन जैसे कि अल्जाइमर डिजीज ही ले लो इसमें अमाइलॉइड बीटा प्रोटीन्स होते हैं जो मिसफोल्ड होकर ब्रेन के सेल्स को ही डिस्ट्रॉय करते हैं पार्किंसन डिजीज है और भी हमारे पास प्रायोन डिजीज है जिसमें कि प्रोटीन की मिसफोल्डिंग होती है तो हाय आई वेलकम यू ऑल टू द टुडेज लेक्चर टुडे वी विल बी डिस्कसिंग अबाउट द structural classification of proteins because structure is as important as the functions of protein. So let's move on to the structural classification of proteins. Okay. So we know that every protein in its native state, in the state in which it is formed, has a unique three-dimensional structure. And this three-dimensional structure is referred to as the conformation of the protein. So the 3D structure is referred to as the conformation of the protein and it is composed of 20 different amino acids. Okay, so whenever it comes to the structural classification, the proteins they are classified into primary structure, secondary structure, tertiary structure, and quaternary structure. All right, so moving on to the primary structure first, जल्दी से समझेंगे और सीधी तरह से समझेंगे, ठीक है? The sequence of amino acid forming the backbone of protein, that means जो primary structure होगा वो आपकी एक linear chain होगी. Okay, so that means the primary structure of the protein will be only held by the peptide linkage. Here you can see एक amino acid, दो amino acid, तीन amino acid, they are together held or linked by the peptide bonds. And similarly, in this structure, you can see that at one end there is carboxyl terminal free, the carboxylic group free, it is known as the carboxyl terminal end or it is known as the C terminal end. And at the another end, you can see that the N terminal end it is free, hence it is known as the amino group is free, it is known as the N terminal end. So yes, the linear sequence of amino acid is known as the primary structure of proteins. Okay. So now why this primary structure is important? Let's have a look. So there are many genetic disorders which I was talking just right now due to the misfolding of the proteins. So these genetic orders or these genetic disorders actually they arise from the abnormalities in the amino acid sequences. So primary structure is most suitable whenever it comes to comparing the structures of proteins to the amino acid sequence. All right, so yes, it holds a lots of value whenever it comes for diagnosing or studying the disease genetic disorders. All right, so we just see the amino acid sequence present in the primary structure only. Okay, now let's move on to the secondary structure of the protein. When the primary structure is being folded and twisted regularly, and the chains are brought in so closer to each other that apart from the peptide bond which was in the linear structure, the peptide bond was in the linear structure and now apart from the peptide bond there is additional bonding in the secondary structure which is known as hydrogen bonding. Now there will be hydrogen bonding in the structure, it becomes a secondary structure. So secondary structure of the proteins, it is holded by two kinds of bonds. One is the peptide bond and another one is the hydrogen bonding. Why hydrogen bonding is so prominent? Because the two chains are coming close in contact with each other due to the folding and twisting. अब एक धागा है, उसी को हम बार बार फोल्ड करते रहेंगे, उसके अमाइनो ग्रुप सामने सामने आ जाएंगे और वहाँ के जो हाइड्रोजन है वो कार्बोक्सिल ग्रुप के सामने आएंगे, तो वहाँ पर हाइड्रोजन बॉन्डिंग हो जाती है और तब बनता है प्रोटीन का सेकेंडरी स्ट्रक्चर. ऑल राइट, सो वेन एवर वी टॉक अबाउट द टाइप्स ऑफ सेकेंडरी स्ट्रक्चर, द सेकेंडरी स्ट्रक्चर इज ऑफ two types. One is the alpha helical structure, another one is the beta pleated structure. Okay, so let us now move on to the alpha helical structure. So in the alpha helical structure we see that the helix is right handed. Okay, it was deduced by Pauling and Corey. Okay, so it is a right-handed helix. This is a right-handed helix. It contains a C-terminal end and a N-terminal end. Okay, so this helix, this is stabilized by the hydrogen bond, and the hydrogen bonds, they are formed between the amide group of hydrogen, the hydrogen of the amide, matlab nitrogen, and the CO groups of the same chain. Okay, and the CO groups of the same chain. All right, the axial distance between adjacent amino acid is 1.5 Armstrong. Okay, the axial distance is 1.5 Armstrong, and there are 3.6 amino acid residues in each turn. And one important thing, that the first and the last amino acid, it will not participate in the hydrogen bonding. First and last will not participate in hydrogen bonding. All right, so okay, so there are certain proteins in which the alpha helices, they contribute to the secondary structure. Okay, but there are alpha keratin, myoglobin and hemoglobin. But there are the structures in which the contribution of alpha helixes is very small, very negligible. आप जिसको कह सकते हैं कि अगर हम काइमोट्रिप्सिन का स्ट्रक्चर देखें तो उसमें बहुत ज़्यादा आपको अल्फा हेलिक्स का स्ट्रक्चर नहीं मिलता, लेकिन दूसरी तरफ अगर हम केराटिन देखें, केराटिन में आपको अल्फा हेलिक्स नज़र आ जाएगा और मायोग्लोबिन में आपको अल्फा हेलिक्स नजर आ जाएगा, हीमोग्लोबिन में आपको हेलिकल स्ट्रक्चर नजर आ जाएगा. ऑल्दो द मायोग्लोबिन इट इज अ टर्शरी स्ट्रक्चर, अभी हम समझेंगे, बट यस उसमें भी हेलिकल स्ट्रक्चर आपके फॉर्म होते हैं. सेकेंडरी स्ट्रक्चर की ज़्यादा फोल्डिंग से मायोग्लोबिन बनेगा. ठीक है, अच्छा. Now, one important thing, that glycine and proline, it is very important point. Okay, glycine and proline, they are the two amino acid that destabilizes the alpha helical structure. Okay, so now alpha helix structure was right-handed helix. Now moving on to another structure, which is the beta pleated structure, or you can say this structure is the stretched structure, है ना. इसमें आपकी क्या हो जाती है, stretching is there. So the surface of beta sheet appear as pleated structure, and these structure are often called the beta pleated sheets. So these polypeptide chains, they are fully extended. They are not coiled as in the alpha helix. Rather, they are fully, extended. And they are composed of two or more polypeptide chains. So now these polypeptide chains, they can be arranged in this manner. You can see here, See, this is the linear chain which contains one N terminal end and another terminal end is the C terminal end. Here you can see that the C terminal ends, they are opposite to each other, whereas the N terminal end, they are opposite to each other. That means they are not in the same direction. Such beta pleated sheets are known as anti-parallel beta pleated sheets, and they have intermolecular hydrogen bonding, whereas the parallel beta sheet contains C terminal ends lie on the one side parallelly, okay, and N terminal end, they lie on the one side, okay. So they are the parallel pleated sheets. So, besides the alpha and beta structures, there are beta bands, there are loop regions and disordered regions, they are also found in the protein. Okay, now moving on to the tertiary structure. So this structure is the tertiary structure. I have not put the heading, so I might write it here. This structure is the tertiary structure. So the tertiary structure means that the secondary structure is folded now again. It is folded, it is twisted. That's why we were saying that alpha helix is also found in myoglobin. But myoglobin is a tertiary structure. Why? Because the secondary structure is further folded about itself, forming the 3D arrangement of a polypeptide chain. So it is a folded, excuse me. So it is folded in such a manner that the tertiary structure is formed. And due to this constant folding arrangement, even the amino acid residues that are very far away from one another, they are brought very near. And once they are brought very near, there is a bonding, even disulfide bonding in the uh, tertiary structure. Okay. Now, the quaternary structure of the protein. So, see, there is a difference that the tertiary structure will be formed by twisting and folding of the secondary structure. But the quaternary structure consists of two or more polypeptides. Please remember this fact that in case of tertiary structure, there will be a single polypeptide chain twisted and bended and folded in such a manner that it forms the tertiary structure. Why? Because there are more and more bonds involved in the tertiary structure. In the primary structure, we saw that there were only peptide linkages. In the secondary structure, additional hydrogen linkages was there. But in the tertiary structure, disulfide linkage also plays important role. Okay, so the quaternary structure, when two or more polypeptides, which may be identical, which may be non-identical, okay, when they are forming a single functional unit, such structure is known as the quaternary structure. Such as hemoglobin consists of two alpha subunits and two beta subunits, so it is the quaternary structure of the protein. All right. So if the polypeptides are two, they are known as dimer. If the polypeptides are four, they are known as tetramer. So now let us move on to the bonds or the forces that are responsible for holding the structure of proteins. So first of all, we already know that proteins are stabilized by either covalent linkage are the linkages which are formed by mutual sharing of electrons, or the linkage may be non-covalent. So when we talk about covalent bonds, there are two types of covalent bonds. One is the peptide linkage, and this peptide linkage, it was between the two amino acid, one amino acid at this side and one amino acid at this side. This is amino acid 1, this is amino acid 2. So the bond between them was the peptide bond. And another one covalent bond is the disulfide bond, which comes into play when the primary structure or the secondary structure, we can say, it is folded, twisted, and bent. Then the disulfide bond will play its role. Okay. Now moving on to the non-covalent bonds. So non-covalent bonds will be mainly of four types. Hydrogen bonds. We know that hydrogen bonds, they are formed between hydrogen atoms and oxygen atoms. Okay. Carbonyl oxygen of the uh, uh, carbon. Okay. Then and hydrogen of the amide, the amino group के साथ में जो hydrogen है, so there will be formation of hydrogen bonds. And apart from hydrogen bond, there are certain hydrophobic linkages also. The non-polar side chains of the neutral amino acid, they tend to be acidic amino acid because they have positively charged group. Okay, so due to this, there is a linkage formed, and this linkage is a hydrophobic pocket type linkage. Okay, water cannot enter due to the nature of these amino acids, so it also holds the structure of protein. Okay, so, last are the van der Waals forces. So, these van der Waals forces, they are certain non-covalent associations, and they are found between the electrically neutral molecules. They are formed by electrostatic interactions. There are either permanent dipole moments or induced dipole moments. So, please comment down how was the lecture, because that's all in this lecture. And in case if you want any other topic to be discussed, please let us know and stay tuned. Thank you.